the thesis, the hypothesis I will tell to you. The first question I want to address is the question, why do we need in pediatric oncology AI? And the reason for that is quite obvious. You can here see from 1940s to up to 2010, and if you would prolong it to 2020, that there was a steep increase in survival rates, but in the last decades, this survival rate is stopping. There's a threshold, and with the common methods, we cannot overcome this kind of threshold. So we do need new things how to solve these problems. Our society is changing over time. And if you have on one hand here the high street banking, local stores, records, printing press, hospital and clinical based care, and you see what happened during the last years, then you see that from high street banking, online banking, local stores to online retail, records to music downloads, printing press to ebooks. And the question is, what is happening with our hospitals and the clinical basic care? If you look for what stands these things for, you see that on the left side, this is all producer controlled. It is a heavy, slow resource. It is information poor, and the consumer has to go to the source. And on the other hand, this is controlled by consumers. It is a very flexible resource. It is information rich, and the producer has to respond to the consumer. And then the question is, what is happening with the hospital and clinical based care from the current healthcare standards approach? What are new standards approaches that will be coming up? And we have to see that medicine is changing. It is changing from phenotyping to genotyping. In former times, we have for the same phenomenological disease, like here colon cancer, one kind of treatment, and this was effective in some patients and not effective in others. Even in some patients, it did even have an adverse event on survival. With the help of molecular biology and genotyping, you now go and try to divide those cancer patients with the same phenomenological cancer into different subgroups. And for each of these subgroups, you develop a specific treatment. And with this specific treatment, you will have a better effect on outcome, etc. So this is what is medicine changing. But how far can we go here with this? Here's another point which I want to stress before I go into other details. As scientists, we have been raised with a view that the best research is hypothesis-driven. But the question is not whether hypothesis-driven research should be one way to conduct science, but whether it should be the only way. And today, the question is about always big data, and the question is data-driven science. And this was addressed also more than now 20 years ago. And this is what we have to look into. And if we have to look into that, we will see that there are examples already existing where big data enable discoveries. And here in already 2017 in Nature, it was published that deep neural networks trained on more than 100,000 images of skin lesions can now classify images of skin lesions as benign lesions or malignant skin cancer as well, and faster than expert. So data-driven hypotheses and discoveries are really getting real. And we want to go with this kind of analysis and research to personalized medicine. And as a physician, then you have the option, go to personalized medicine, or again, one size fits all. And I think we need to go to personalized medicine. I just show you one case of a 9-month-old girl who was diagnosed with nephroblastomatosis on both sides, so huge kidney tumors on both kidneys in this small child. This child was treated according to the protocols that were available at that time. This is more than now 10 years ago. And the tumor did primarily shrink on both sides, but then it grew again. And that's you see here. And it was as large and huge as at the time of diagnosis. And the question was, what to do with this child? Should we take both kidneys out? The child has to go to dialysis, needs a kidney transplant, and this in a 9-month-old girl, it will affect the whole life of this child. And what we did at that time, we did look into the molecular biology and found out that this child had disruption of the retinolic acid pathway. And when we in vitro gave to these tumor cells retinolic acid, we could see that the expression of the different genes in this pathway did normalize. So it was something that happened in the lab, and we thought, okay, retinolic acid might be a possibility to treat a child because the side effects are low, and we do this kind of treatment already in children with neuroblastoma. So this child did receive, here you see again, this was the time of diagnosis here, and you see the treatment with vincristine and actinomycin D chemotherapy, how the tumor volume went down and went up again. And at that time, we started with vincristine, actinomycin plus the retinolic acid. And you see the tumor did go down. We stopped retinolic acid. We stopped the chemotherapy here and continued with retinolic acid. And the question is, what did happen? You see here diagnosis, again, growth of the tumor, and then start with retinolic acid here on the far right side. And you see the kidneys went to normal with no tumor anymore in the kidneys. The child was never operated. And this is now more than 10 years ago. And the child is still living with 2 normal kidneys and is healthy. And this is what we want to achieve in all these kids, that they will get a treatment that will cure their tumor, and they should not have long side effects of the treatment or the tumor. And this is much better than having taken out these 2 kidneys at that time and then going for transplant and lifelong immune suppression for the transplant. So that means in all our kids with nephroblastoma, we have achieved with conventional treatment really a good success story, as I have shown before. Most of our kids with kidney tumors, and especially with nephroblastoma or Wilms tumor, will survive today. And we can, for the whole cohort of patients, try to find risk factors. And if we, for example, look into those patients according to their treatment they do receive, there's a part of patients who receive low treatment with only 2 drugs. Others receive also 3 drugs or radiotherapy. Then there are high-risk patients with 4 drugs. And there are stage 4 patients, meaning patients with metastatic disease. And you see here the number of patients, and the majority are treated here in the low-risk group with vincristine, actinomycin only. And they have a survival rate of 90%. And the survival rate is going down, the higher the risk is, and the more the treatment is given. But now you can think about, okay, this is fine, and this is really poor. So increase treatment here, and maybe go down with treatment here because there's a high survival rate. But if you look to the absolute numbers of patients that do relapse, most patients do relapse in this low risk group. And there's up to now no findings why are here so many patients are surviving. And they have about 38% of all relapses in this group. Whereas in all the other groups, the percentage is lower. There's still a high number of relapses in stage 4. But what you can see here as well, if you are looking into the death rate, these patients can mainly rescued, and they are only 14% accounting for the death rate, whereas in stage 4 metastatic tumors, there are 50% of patients who will die. And the question now is what to do with this data. It is not the solution that we will here go down with treatment and go up with treatment, because we need to find those single patients that will have here poor outcome, despite the fact that with all the known risk factors of today, they have an excellent outcome. And there's another question. In our last nephroblastoma trial, where we had randomized one drug, namely doxorubicin, which causes late effects to the heart, and asking if this drug is really necessary in patients with a stage 2 and 3 and having a specific intermediate risk histology. And what we did find out, we can, looking at the overall survival rate, we can really take this drug out of the treatment protocol because patients with and without doxorubicin are doing the same way, and the overall survival curves are superimposable. If you look into the event-free survival, then you see there's some small difference, which is not statistically significant. But the question is, do we want here to reduce the number of relapses, or do we accept some relapses more in those patients not receiving doxorubicin? This is an ethical question, because to spare one relapse, we can calculate that we need to treat 20 patients with this drug and having late effects to the heart to spare one relapse. But all the relapses that occur here can be rescued with a relapse treatment. So there was no reason here to really let doxorubicin to be given to all the patients. Nevertheless, there are two questions coming up in this trial. The first question, how can we define those patients that do relapse when they only receive 2 drugs? And can biomarker for risk stratification help to close this gap of event-free survival? And even if we have here a high survival rate of more than 90% of the patients, there are still patients dying. And the question again is, can biology help us to close this gap and find those patients who do need another treatment not to die on their tumor? So then I want to ask you, what are the challenges of AI, especially in pediatric oncology, but it's also challenging in whole clinical medicine. What we all know that we need data, a lot of data. And with these data, we will drive our new knowledge. And the questions we have to ask are about the availability of the data. How can we standardize data? What is the semantic interoperability to data? How can we share data? And this is especially true for pediatric oncology, how big is big data? You do know that pediatric oncology and all the different diseases or cancer types in pediatrics are rare tumors. And if you have a rare tumor, how can you go to big data? This is a really very important question. And you need to think about the quality of data as well. And if you develop with all your modelers and with your team AI models, the question is about explainability and how can clinicians trust these models? Clinicians are not knowing about the algorithms behind the models. This is a black box for them. And if they should use it, then they need to trust these models. Then we have another really big issue in artificial intelligence. This is the questions of regulations. There are legal regulations, ethical, and other regulations. And you have the medical device law, which I believe is something, this is so important if you have set up a model, and then there comes the regulation of a medical device law. At least in Europe, this is somehow very difficult, and from academic perspective, you cannot solve this problem. And then there's the question of interdisciplinarity. It is very important to know that you can only achieve good results if you work together. And working together means you need to put all the stakeholders together, and they need to understand each other's. To see what you can achieve together, and they need to speak the same language. If you are looking into the large scale of data and you want to compute it, that starts with the molecular data, über immunology, cytogenetic data, histopathology, imaging, segmentation data, functional data, then you have the clinical data, and that may end up with epidemiological data. And you can also think about social media data. There are so many kinds of data that can be used, and they are heterogeneous, and you need to think about how to bring them all together and analyze them to get a very good model that you can really have a better kind of going to precision medicine or personalized medicine by using these models for decision support. There you have the frequency here as a kind of questions how molecular biology in now nephroblastoma may help you. And if you just look into one marker, you see you find these marker of copy number aberrations of 1q gain in only 28%. And this is the highest number of all these different CNAs. And you find 45% with no markers. So just one marker is not helpful for you to find it. And especially if you look into molecular biology and try to put them together in live tables, you will see that you cannot define a big difference in those having the marker and those not having the marker. And this is for event-free survival as also for survival. There are only a few where you see here a big difference, but these are very rare situations. And this makes it really difficult, and you need to find more and better markers, and it's not done with only a single marker to get a better outcome for patients. There's another marker which is coming from histology. As you may know, that nephroblastoma is a disease that is very heterogeneous in histology. There are epithelial parts, stromal parts, and blastemal parts. And we do know that a part of the blastemal tumor cells will not respond to chemotherapy. As in SIOP, we treat them all preoperatively. We can also measure the amount of blastemal that will survive the preoperative chemotherapy. And here, these are patients with stage 4, meaning with metastatic disease. And you see here in this plot the number of patients and their relapses. And these relapses are going up if the amount, the absolute amount of plastemal volume is more than 10 milliliters. And this is also shown in live table, showing that if you have a stage 4 metastatic disease, and the only risk factor which is independent from other risk factors divides this patient group into two entities, those with less than 10 milliliter of plastemal tumor volume and those with more. And this difference in outcome is just based on volume of plastemal cells. But there are still patients who will not survive if they have less than 10 milliliters. And there are patients that will survive despite the fact they have more than 10 milliliters. Even if there's a big gap between these two groups, it solves not a problem to find for each of the children a solution. And the question is, how can we find all those data we would need for our AI models or all kind of models we want to use as decision support for pediatric oncology? And there are 4 questions I have listed here. The first one is, how do we know what data we have? Are we always sure we have all the data we want to use? And how do I know what data you have? Because we want to share the data, and maybe this is helpful to get data from somewhere else. And how do we know what data we don't have? And how do we make different sorts of data combinable? So these are questions that need to be solved. And we need to think about that. How is that possible? One answer for that is the international collaboration, especially in rare tumors, that you need to work together to find kind of data. For example, if you have nephroblastoma, and in whole Germany, we see only 100 kids with this kind of tumor each year. And we do know that 90% of these kids will survive with standard treatment, and only 10 will die on this tumor. And to find solutions for them, it is necessary to work together. If we are looking for clinical data, it is still the case that most of the clinical data are paper-based. They are not in the computer. And the question, how to get these data into the computer? You need to think about it, and you need to work with people that will help to put these data into the computer, or you need to think about computer systems where you've directly put all the data in the computer system. There's another thing which is important about data. This is the quality of data you have. And the quality of data, here's one example we have used in or analyzed in Germany, where we looked where are these patients with nephroblastoma are treated. And as it is a rare tumor, we have divided our hospitals in 3 kinds of tumors, of numbers treating these patients. There are only 8 hospitals in Germany treating more than 4 patients a year. And you can ask, how experienced are those hospitals? And these are half of our hospitals treating less than 1 patient a year. And you see, if you count the numbers, you see this is just 13% of all patients, whereas here most of these patients are treating 1 to 4 patients. And the question is if they do and behave the same for patients. And if you look into one single question, here you see the numbers and the hospitals, and just look into what happens in those hospitals treating less than 1 patient here in red bars, and those treating more than 4 patients, you see that in surgery, you have here a higher percentage of patients where the tumor did rupture during surgery. And this is not because they have the larger tumors in these small hospitals. No, they have the smaller tumors. the less patients with risk factors, and despite that, they have a poorer outcome of surgery. And the experience and the quality control of treatment is important, affecting the outcome. And how can you put these data into a model? This is somehow very difficult to answer. And then there's the question of reproducibility and credibility of the data you will collect. And that starts with the sampling of the tumor. Do you have normal tissue when you sample tumor, or is it really tumor? Do you have a reference tissue to compare tumor against normal tissue? Do metastases have the same molecular biology than the primary tumor? And we do know in nephroblastoma, and I'll show you that later, there's an intratumoral heterogeneity. The question is also not dealing with the tumor alone, but also with the microenvironment. Who is looking into the microenvironment? Then there are questions about the processing of the tumor samples before you analyze them. What is the time frame? Do you freeze them? What kind of transportation do you use before you analyze them? Which method do you use? What are you doing with the raw data? And then you have tools to create hypotheses and knowledge from data and reports. And how is that reproducible in all these cancer types to find the correct data? And here you see one kidney with a tumor inside after surgery, sorry. And what you see here as well, there are several areas of the same tumor, and here the normal kidney, where you can take some tiny specimens to analyze for molecular biology. And if you look just for one Q gain in these tumors, and you have here different patients where you have more than one sample analyzed, and then you can look into the question, how often do you find a tumor sample with one Q gain, and how often you don't find it, despite the fact that it is the same tumor in a single child. And now think about, you would look for this sample, these samples where you find the tumor, the one Q gain, you can find it in 100% of patients, the one Q gain. But if you have just these samples, then you only find it in 3 patients. And how can you define that one Q gain is indeed a marker for tumor outcome? And this is somehow where you need to think about what kind of data do you have? What is the quality? What is reproducibility? And the question is, can you change that by other kinds of collection of data? And then you go to the question of circulating cell-free tumor-derived DNA to try to reveal the genetic landscape of Wilms tumor for diagnosis, response, and follow-up. As you think that if there's a tumor in a kid, it may release tumor cells to the blood. And if you analyze the blood, you may find in molecular analysis DNAs for different molecular markers. And that may give you a better overview of the molecular biology of this tumor. But still, a tumor in a child is not just a tumor. There's also the host and the immune system reacting with the tumor. And do you collect also data for the immune system and the host, which may play a very important role to overcome resistance of tumor? We have done a microRNA analysis in the blood of some patients and tried to find out if also the immune system has a specific impact on outcome. And we did find that specific microRNAs are corresponding with the response to treatment. Specific microRNAs are correlated with tumor necrosis after chemotherapy and others with more progressive tumor cells, with plastemal tumor cells, and also with tumor cells where the proliferation will be enhanced and apoptosis is stopped. So we need to look not only in the tumor itself, but also in the molecular biology and in the immune system of a patient. And here's the Tower of Babel. Why do I show this picture? I show it because, and you can see here, to understand data that are in the computer and that computers can talk to each other, that you need ontologies, you need to think about standardization of the data. And here, if you have the warning of bears, and you see here the family going around, there's the teddy bear, he is angry about the bear, and she lies on the fireplace on the bear. So this is the same word, but different meanings behind. There's not a very important issue. If you have a clinical trial or if you have a patient, this patient will have a time course. And if you have the data only at one time, you may lose really the whole time course of a patient. And here, the question of curation of data is important. Here you see, if you have here 3 different treatments for the same tumor type, the treatment 1 is doing better, and it is doing significantly better than treatment 2 and treatment 3. So if you would stop here the treatment, the registration months, 60 months after diagnosis, you would advise give all patients the treatment 1. But if you follow up these patients, it may happen the following. There will be no further relapses in these 2 patient entities, but all those who have survived here up to 60 months will increasingly step down. And after 108 months, treatment 2 and 3 are better than treatment 1. And the question is, which treatment is the best one? And you again have here cohorts of patients and not a single patient. Who tells me why is this patient here at that time relapsing or dying? And why are these patients here doing better than those patients here after a longer time of period? This is not helpful in finding better treatments for patients. And you need also look into patients-related outcomes. It is not always the question of surviving, but it's also the question of how to survive. Then if you are looking into data, you always have a value conflict. This is the question of privacy of the data and access of the data. This is in the individual interest of a patient, and this is in the collective interest of the society. And this conflict between privacy and access to data must be balanced. And here is the question how we can deal with that. In several European projects I was in, this was a question always that did hinder us to receive data from other places. And we did set up a legal and ethical framework that demonstrated us also showing to the European Commission that this data protection security framework will make data a kind of anonymous. They are not anonymous, but we call it de facto anonymous, as you do need a lot of work to really get back to the original and private data. You all do know that molecular data you cannot anonymize, and imaging data you cannot anonymize, and so on. And this data protection security framework is based on 3 pillars. The first pillar here is the informed consent and the ethical approval. So you need to get informed consent from your patients and you need to get the ethical approval. The second pillar is the secure de-identification and with technical safeguards. You can pseudonymize the data technically, etc., but both together will not de facto anonymize the data. And there's the third pillar we use now in our European projects. This is a network of trust and contracts between the data provider and the data user, so that you can only do this kind of research with your data that you have written down in your contracts. And if you are doing more, then you will get into problems. So such a legal and ethical framework is very important to have. But these regulations that came up are also showing another problem we are facing. And this is the problem, I should show it with the renal tumor study group and the different trials we had here over time. And we started with a trial in the beginning of the 70s in the last century. And what you can see here, that the number of patients is increasing from one trial to the other, because the outcome is getting better and we do need more patients to show in our randomized trials the benefit of new treatments. This is one point. The other point we are facing is the problem that we need more and more time to start with a new trial. At the beginning, we ended one trial in the same year when we started the next one. And if you see here, there were 2 years between SIOP-9 and SIOP-9-301 before the other trial could start. And therefore, we did prepare the next trial already early so that we could start early. And this trial stopped in 2015. And the new trial has just started now. And the reason why it did start just now is because of the different regulations you have to face. And as we have an international collaboration, because of the rarity of the tumors, it gets more and more difficult dealing with the clinical trial regulations, with the data protection regulations, and so on. This is one of the reasons why I strongly believe that all these regulations are not helpful in finding new and better treatment for patients. If we would have these regulations at the beginning of the 70s last century, we would not have increased survival rates of our patients as we have done before. And you see here, this trial with just 2 centers, one in Amsterdam, the other one in Paris. And now we have in SIOP 2001, 28 countries from 261 centers. Today we have all countries from Europe. We have Brazil with us. We have countries like Egypt, South Africa. We have Singapore, Hong Kong, and some centers in China like Shanghai, Guangzhou. Japan wants to join us. So we have a very international study group now to really put all these data together. And the more we put them together, the more we have to look also on the quality of data. But there are also other legal consequences. When we have set up an AI model, then the question comes up of trusting the AI model. And the question we have here is the question, is the AI model predicting a correct treatment, yes or no? And if the AI model is predicting a correct treatment and the physician treats according to the AI model, everything is fine. And if the AI model predicts not a correct treatment and the physician treats not according to the AI model, it's all fine. But what happens if the AI model will predict the correct treatment and the physician says, no, I don't trust this AI model. I treat the patients according to my standard. Then the patient will have a poorer outcome. And the other way around, the AI model predicts not a correct treatment. The physician says, oh, this is modern treatment. This must be correct. And I treat my patient with what the prediction tells you. So what we need to think about, how do we solve this problem? And one point is that you need to really show that an AI model predicts a really correct treatment. There's another point which is important, and many people are not thinking about that. If you have a patient with a new tumor just diagnosed, and you want to use an AI model for his treatment, then you need the results of the AI model in due time. And if you need a lot of data to build up the AI model, like imaging data, biomaterial to analyze, clinical data, and other data, there's a pre-processing of biomaterial and of imaging studies before they can go into the model. And then the model will predict you how to deal with the patient. And you see, you have here a timeline. And before you can start with the treatment, you need to look into the time constraints. You cannot wait until 4 weeks and say, okay, I need this time to get here a result from my AI model. No, you need it earlier. Otherwise, you cannot use it in the clinical setting. And these time constraints are very important. There's another thing, if you want to get clinical data and molecular data, imaging data, and something else to really use them. Many people are saying, these are my data. I think it is important to show that all those who share data should be acknowledged. You need to thank all those who helped in carrying out the research. So in your publications, data sources should be cited in the references sections of user publications. And the data user be a sole responsibility for recognizing the role of the data provider. This becomes very important to acknowledge the data sources and the providers. It is not enough to cite papers from other people, but you need to give acknowledgement to the data you have received to run your research, to get new knowledge and to write a new paper about your research results. And it is always teamwork. I have told you, you cannot do build up an AI model without having a good team. And you do need IT specialists, you need other scientists, you need the molecular biologists, you need clinicians, and you also need lawyers and ethicists. Otherwise, it will not work. And if they work together, you may be able to build up a kind of team that will drive you to a better way to get results from AI research. So you need to set up an architecture. If you have a legal and ethical framework, you need to have the data, you need to have tools that you are developing. They need all to be interoperable. Then data needs to be anonymized. There will be a workbench where you can test all your tools you are developing. You do need workflow repositories. You need a coordinator for that, and everybody needs to work together. And then if this is teamwork, then the train can drive to personalized medicine. And this is how you have to work together. With need a data warehouse where you may get the possibility of uploaded data, but you need also the patients that allow you to upload the data, and patient empowerment is very important, so that the data upload is better. Then you need query tools to get access to the data, and you need an authentication of those who are allowed to do so. And the other way around, not to have a data where, and this is also important that you do data curation, as I have shown before. And the other way, not to have a data warehouse, is the question of federated data research. So you always go directly to the data and then do the model analysis on your computer, so that there's no collection of data necessary. This is somehow that needs to be discussed in the infrastructure that are developed. Now I will go to one work that we are trying to build up a model in nephroblastoma. And what is important for me, and I want to stress that here, is if you are trying to build up models that are really interesting in clinical perspective, it is important to think about a relevant question. If you do not have a relevant question from the clinical perspective, and you are not working together, this will not be helpful to build up models for clinical decision support. That means also, again, you have to work together as a team. And here is one simple question we are asking in nephroblastoma. As you may know, in Europe, in SIOP, we always start after having the diagnosis of nephroblastoma with preoperative chemotherapy, without having the histology. And the question we are asking in a specific child is the question, no, why is it not going further? Is the question, sorry, is the Wilms tumor of this patient shrinking after preoperative chemotherapy? This is a simple question. We do know that about 80% of all children with Wilms tumor will have a tumor that will shrink under preoperative chemotherapy. Only few will increase under preoperative chemotherapy. And the question we have, can we answer this at the time of diagnosis to select those patients who really do need primary surgery because the tumor will grow under chemotherapy, compared to those who can be shrinked under preoperative chemotherapy, make tumor surgery much easier, getting a downstaging of the tumor, which will need less treatment after preoperative chemotherapy. This is the question we want to answer. And for that, we need data from the patient. We will have the model to execute and visualize the results, and this will lead us to decision support in the patient. So the question is, do we need data in the data warehouse, the data models, put them together and validate them, and then to have the decision support? From a clinical perspective, there are two important areas. The first area is the question of data management. So how to get all the data together in due time, meaning also pre and post-processing, uploading of data, pseudo-anonymization the data, etc. And then how to execute the model. What are the data we do have? So we have here the patient with a specific tumor, and we have the question. We have blood samples from the patient, we have the clinical data, we have imaging data, and from the blood samples we may create also molecular data, and from the imaging data we may get other kind of knowledge. For example, we have here the question of the ADC values and cell density, which will show you not only if the tumor is shrinking during chemotherapy, but also if cell density is going down or up. This can be done with diffusion-weighted imaging. We can look into blood perfusion, looking into the imaging data, etc. And we may put all these data together with other models that are available, like pharmacokinetic models of the drugs we want to give to the patient, etc. And this model will be done for each single patient and will give you an answer if the tumor will shrink or will not shrink. And as you have heard before, for that we do need collaboration with different stakeholders. Here, the starting of this project in a European research project, and you see here different partners from different areas in Europe, and even here the University of Pennsylvania and Philadelphia, who did participate in this project and to set up this model, which we call a multi-modeler hypermodel, because we have a granularity of models here, vascular model, a metabolic model. We have the oncosimulator, which is primarily looking into the growth and response to chemotherapy. We have a biomedical model, biomechanical model. We have a pre-processing tool for specific data. And we have here the molecular model, which is looking into the microRNAs and other genetic data from the blood. And this tumor model was put into the computer and shows the tumor at the time of diagnosis in visualization mode. And you can use that. And at the end, you will see how the tumor volume will go down. And the tumor at the time of diagnosis, and the tumor looks after the preoperative chemotherapy. And this is put into a clinical research application framework. And we have not only looked into nephroblastoma, but also in other tumor types as non-small cell lung cancer, glioblastoma, and prostate cancer. And we have built up this tool, which is now refined again. And we have here the nephroblastoma hypermodel, and we will show you how it looks like very soon. And you can select the data there, and then you will get an answer when you run the model. And therefore here, it looks like, as I show you here in this short video. So you have your credentials for the clinical application, and you start this clinical application for a single patient. Then you select your tumor entity, and you will answer a question. This is the question we have asked here. If the patient responds to preoperative chemotherapy by tumor shrinking, then you can select the patient. These are fake patients here at the moment because of privacy issues. And then you have the different data types here. You select all these data types. You select the model that you want to use. And then you press button, and then you can put here the kind of chemotherapy you apply to the patient in these areas. And then you can also expand to other data you will receive from molecular analysis and from others that are automatically uploaded for this patient. And then you can execute that. That takes some time to execute. I hope this will continue now. Now I will show you. So you have here different runs of this model. And then you can select the model when it is executed. And you can have a 3D visualization of the model and the outcome of the model. You select the data of this model, and you see here in a three-dimensional way how the tumor looked at the time of diagnosis. The tumor was automatically segmented, And over time, you see here how the tumor is shrinking over time. And you can put all this in a report. Select the time of diagnosis and maybe selecting the time after half of the treatment. And then you put this together with the end of treatment visualization of the tumor. And then you receive a report that you can store and upload, create a report. You can upload it to the database you have. And this report shows you how the tumor is shrinking over time. You see here, when you give treatment and the tumor is going down in size. You have in your report also the images that you did select before. You have here the different hypermodels that are put together and all the data that were used for this specific patient. And this is a tool that we are now refining together with the Technical University in Athens, with Professor Stamatakos, and to run this model now for many patients to optimize this kind of model, so that we then use this model. And you here see, if we have other parameters, we will get other outcome of the model. Yeah, the result is different, and this shows you that there is a validity of the model, and we can always compare the outcome of the model with the ground truth of the data, as we do know how the specific patient was responding to the tumor or not. This is the example how we set up a clinical model for nephroblastoma. And now I need to see how it is going on. Sorry. Okay. And if we look into artificial intelligence, specifically in pediatric cancer, what is available today, we must confess that research on AI in pediatric cancer is still in its infancy and is not translated into clinical routine yet. The most available studies rely on small and not representative data sets. And most published studies focus on radiomics and brain tumors. This is a result from another European project. We are just looking into AI in pediatric oncology, and this shows us that there is not a lot of other things done. And many registries exist in the European Union, but they don't seem easily interoperable. And ethics in AI for pediatric cancer is still immature. And there are no efforts yet on telemedicine and AI in pediatric oncology. So there's a real great possibility of making this come live. And if we are looking for potential areas of activity for artificial intelligence in pediatric oncology, is also a result of one of the ongoing European projects. We can look into different ways there. Here's radiology, radiomics, digital pathology, genomics, clinical decision support, telemedicine, and what is done here in screening, in non-invasive diagnosis, in prognosis, precision therapy. What data are we using? And you see only where it is colored here in this yellow. This is somehow where you can get something to think about if that is going on. But then what every clinician needs to have is trust into the model. And how can you trust in a model when you do not understand the black box? Data are going in and the result is coming out of the black box. What clinicians understand is clinical trials. if a new drug is going to the market, it needs to go through different phases. And phase 3 of a clinical trial is the randomization between a drug A and a drug B, where you do not know what is better, drug A or drug B. And the question is how to solve that. And this can be solved in clinical trials. And the question I'm asking now is the question, can we use an AI model or another model and use it as a new drug? So there would be the design of a randomized clinical trial where you will get and need ethical approval, where you do randomize after diagnosis and having the result of your model that you treat half of the patients according to the standard treatment and the other half of patients according to the AI prediction. And then you can see what happens here in the standard treatment. They all receive preoperative chemotherapy. And those patients who are treated according to the AI model, only those that the AI model shows a tumor shrinking. And those where the AI model is not predicting a tumor shrinking, you go to primary surgery. And the question is, which pathway here, which part of the trial is doing better? And you have inputs where you can look into the outcome, the stage distribution, the histology, etc., etc. And then you may choose if the treatment according to AI prediction is better than the standard treatment, then you can use this as a new standard. And this is what a clinician would understand. So use these tools that you are developing as a kind of a drug and then try to run such a clinical trial, which will not be easy because as soon as you use an AI model for clinical decision, this AI model is underlying the medical device law, and this is again a nightmare. So this is a lot of questions that are not solved yet. And now I'm coming to the end of my talk. It is not only the question of surviving, but it's also the question of how these kids do interact with their disease. And here is a picture of a child that was just diagnosed with leukemia. And the child did paint this picture where the rain is coming, the sun is disappearing despite the rainbow. And here is the child living in his house. Then the child was treated. And at the end of treatment, the child was cured and survived. And the sun is coming up again. And the sun is putting all the rain and the clouds away. And flowers are growing again. And this is what we want to achieve as well. Not to cure the patients alone, but to cure them in a way that they are feeling quite normal and good. Thanks a lot to all the support we had for doing this kind of research. And thanks to you for listening. And I'm open for questions now. Thanks a lot.
Norbert, it won't be a surprise that I have a question, but I'll try not to monopolize it as I sort of have access to you at will by your good grace. But thanks for those pictures from the child at the end. It always tugs on my heartstrings to see those, and you know that.
I know that. Yes.
A more strategic question is that. Yes. It's not a surprise that my knowledge and experience with AI is aligned with all of the scientific architecture that you have exposed in the collaborations. One of the questions I have is really about international collaboration, and it's not about scientific collaboration because we've been precipitating that for many years. It's about the education of our public systems to invest the resources it takes to expedite this kind of work, because we sit from their point of view, from the politicians and bureaucrats, administrators' point of view, we sit on a rather flat surface, and we need to make the case that this is of higher priority than something or else. And you and I both have experience doing this in Asia, Europe, and me in North America. So the question is, what do you think the most important thing to do is to motivate decision makers to provide accelerated support to achieve the things that you've mentioned? I mean, one thing is to have Norbert give this talk everywhere several million times, but that's not resource efficient. What do you
Yeah.
suggest?
I think the first you have to do, and this is what we are doing in the moment, is to convince our societies so that they will have direct contact to the politicians. And we have with our SIOP a really good contact now to Brussels. And we are setting up now a kind of strategic field for AI in pediatric oncology. There's one European project I'm in dealing with AI in pediatric oncology, and that will be followed by a success project. And this project will have the impact of our societies to the politicians to speed up this process and to convince them that it is needed. This is one very important issue. The second one is to talk to parents group in pediatric oncology, to talk to survivors, and to put these people within your projects, because they want to get a cure as fast as possible. And the third and last thing is that you need really, and I think this is one of the experience I had in the last years, which is getting much better, to convince some of these people that are working in AI or in building up and setting up infrastructures and the usage of data, that you need to convince them that you need to start with simple questions to answer them, to show a benefit directly, because this will influence the attitude of physicians about AI technology. And the last point is that I believe we have to change our medical schools and the education and curricula of our medical schools, because people, doctors or physicians in the future need to know about AI or about these kind of infrastructures. This is important because they will come. And if they do not understand, then I think they will lose their job. Other questions?
So if there's anyone has question, could you just use the raise hand function, and then we can unmute you. But I have a few questions. There's one of the main focuses of my research, but lots of people's research, is the late effects of childhood. And you touched on it a little bit with cardiotoxicity, but is there work being done for predictive models for late effects and cardiotoxicity for ones? Is that sort of one of the objectives that you've been working on, and we'll talk a little bit about that? I mean, not just try to predict which patients are going to be at risk for the late effects.
Absolutely. This is a very important question. And this is dealing with the fact that survival is not all we have to look for. We have to look for the kind of long-lasting side effects. And we all do know, even if we cure children with cancer, their long-term outcome, even if they are cured, is not as good as children who had never had cancer. And we do know after 40, 50 years, they will have a lot of trouble and problems, and that may, and we need to look into that, and we need to prevent that. We have submitted just a project to the European Commission, hoping that they will help us to find these risk factors on individual basis. Who is getting cardiotoxicity if it is treated with cardiotoxic drugs? Because not every child will get these cardiomyopathy. And this is somehow where we are hoping that it will get funded. And we try to get here all the data that are available for these patients to put them together. From the ECG, the echocardiography, in some patients we will also have MRI, etc. And I hope that this will answer some of the questions of cardiomyopathy. But it's not only cardiomyopathy, there is also the question of renal failure in nephroblastoma. There's the question of hearing problems, and so on and so on. And we need to have really a good view on that. And I think this is really important and isn't a field that we can and need to address with AI technology.
I can't see if there's any other questions. I don't see anybody's hand up. So I'll, if it's all right, I'll ask another question.
Yeah.
The models that you generate with your AI, or have generated or plan to generate, how did you validate those models before you know, the ultimate test is the RCT, but how maybe I missed it in your talk, so I'm sorry, but how did you validate that? Was that in a prospective cohort study, or?
Yeah, sorry, yeah. This was, we have collected many data of patients from imaging, etc., and this is a retrospective cohort. And we have developed this model with the retrospective cohort, and there we have, for example, the imaging studies at diagnosis, and we have the imaging studies after preoperative chemotherapy. And these imaging studies after preoperative chemotherapy are the ground truths to validate the response to treatment in the model. And then we can see, and this is how we want to refine the model, is if we have a discrepancy between the prediction and the ground truth in a single patient, then we try to use machine learning to optimize this model. By more patients are running through the model, we think the better the validation will get, and the better the model prediction will get. And this is just on way what we want to do now, together with the university in the Technical University in Athens, to set up a better new version of this model. And this is based primarily on the retrospective data. And then if we have this done, then we will use it in prospective data to see if we can confirm that. But we will not take any decisions out of it, but we want to confirm with prospective data if the prediction is as we expected.
So you're generating the model on retrospective data and validating it on prospective data.
Yes,
so,
right.
Got it, okay. Are there any other questions?
There's a person with their hand raised. There's two people with their hand raised. Can you not see them, Leslie? There's
I can.
okay, so Jake, you go first, then Mike second.
Cool. Okay, yeah.
Thank you.
Thanks for, that was like a really great, interesting talk. I really appreciate it. Thanks so much. I'm curious, like I love how you like mapped out all the potential areas for AI. And I'm curious if you have any ideas about which ones are either like currently furthest ahead, and it sounds like it might be what you're working on right now, and then also which you think might be the latest to develop and actually end up in clinical practice.
I think one important issue is to start with questions that are easy to answer and that may have an impact on the clinical situation. And this is depending on the, for example, cancer domain or what you are looking for. So there are so many questions you can answer and try to answer with AI, but I think the most important would be to be as easy in the questions you are asking. That may convince much more the physicians who have to use it later on. You need to keep in mind, you can try to explain AI models to the clinicians, but most important is they have to trust the AI model. And you need to convince them to trust them. Explainability is important, but it's not all. The more you have trust in your model, the more it will be used.
Cool, thank you.
Yeah, thanks.
Okay, Mike, thanks. And then Randy. You're muted, Mike.
Yeah.
There we go.
Sorry, too many mute buttons. Can you hear me now?
Yes, yes.
Yeah, okay, great. Thanks so much for the talk. Really fascinating. Lots of stuff you've touched on, obviously. I come from Roche, interesting. I think one of the assertions I've been making about AI as I talk to lots of people about it in the healthcare world is that one of the many promises of AI is not just these better personalized outcomes, but ultimately it hopefully will reduce the cost of delivering care and achieving those outcomes. Is that something you agree with, or am I making potentially wrongful assertions,
No.
given the level of personalization and the complexity of bringing the technology forward?
I would agree to that, and I would give you one argument. If AI models will really go to the point that we will increase survival rates, we will have less relapses. And each relapse is very costly. Mm-hmm. And each relapse is, at the end, many of these patients will die. So you will put a lot of money in relapse treatment with no effect at the end. And if our AI models will prevent relapses and will increase the cure, you will spare money. This is one point. The second point is, if you are thinking about targeted therapies, then you can also think about who is really eligible for targeted therapies and not to spread it to everybody. We need to be careful because this is expensive treatment and it should not be given to everybody who thinks that will help him. We need to find ground truth for that. Who is really someone that will be, who will have benefit from such a targeted therapy?
Great.
Does that help?
Yeah, absolutely. And of course, there's obviously the complexity as you figure out the pathways to bring this technology forward to market, reducing the ambiguity of what those pathways look like. Increases the viability of the technology as well.
Yeah, right.
Hey, Leslie, am I next?
Yes, you're next. Thanks.
Okay, thanks. I wanted to make a comment that I know Norbert already agrees with because of our collaboration, but it's to address an issue raised by Jackie, and it's kind of confirmed by Leslie's question about the confirmation of the models on retrospective and prospective cohorts, because it's important. So the first thing I want to say is I don't want anybody to misinterpret Jackie's question as asking questions about AI methods. Don't ask. If you're a physician like Norbert, your subject matter expertise shouldn't be questioned, but you shouldn't be questioning the subject matter expertise of the AI experts. And I get this all the time with half experts telling me what the best AI methods are to do personal medicine. It's Norbert knows this and I know this. It's a big mistake, because what you often end up doing is having half knowledge of the AI science methods. Concentrate on what you're good at and build the collaborations. That's really important, and it affects everything throughout what Norbert outlined, and that is all the way to data gathering. Is I can help you gather better, more valuable data, but I can't and don't have the expertise to decide and distinguish between what's more valuable and what's not.
Yeah.
And that's extremely important. So the minute I see a clinical expert telling me we should use deep learning, be careful if I'm in the room. The same should be said when I question Norbert's best practices on reducing the size of a neoblastoma. Who should ask a computer scientist? No one. So that distinction has become really important, because as Mike Bales pointed out, is that the acceleration of the combination of the data scientists and the subject matter experts clinically are together producing new hypotheses about treatments and new efficient architectures for making suggestions and providing decision support. But don't confuse those.
Yeah,
So
I
Jackie's
completely
question
agree.
helped do that, and Leslie's question about validating models with retrospective and prospective models is important. And I get myself offside with my friends like Sandy and other clinicians when I say things like, maybe we have to think the old-fashioned view of randomized clinical trials. Instead, talk about timeframes of prospective and retrospective cohorts and slide the timeframe window using AI methods for continuous improvements. So we're still doing all the same things, but I can expect to canvass Norbert's program tomorrow, and if there is a better result, it's available. Anyway, end of sermon.
Thanks, Andy. I completely agree.
Ramit, did you want to, you had your hand up?
Yeah, I wanted to follow up something which is I'm curious how using AI models you are to kind of avoid the
You have a lot of feedback. We can't hear you. I don't know why there's feedback.
Oh, I'm not sure.
Try toggling your microphone and then get closer to it. That will improve it a little bit.
Is that better?
A little. If you speak slowly, like you're speaking through a glass of water, then we'll be able to get your question. And now you're muted, so.
Except it doesn't show mute, so I think you have an audio visuals. I think it is time's up, though. That's the only thing.
It is 1 o'clock. So, so maybe we should close the session. And thank you so much for that. It was an incredible presentation, and
Thanks a lot.
it was it was really well, amazing.
Yeah.
So
you fulfilled the expectations, Norbert. I don't think I oversold you.
Okay, thanks.
Thanks.
It was a pleasure for me to give this talk, and I think it will be great to come again. I did like Edmonton, and wonderful. But I have to fly back tomorrow, so time is passing. And thanks a lot for the invitation and being here.
And thank you. It was wonderful. Great.
Thanks.
Thanks.
Bye-bye.
Bye-bye.